Okay, so today we're going to continue looking at metabolism, and I went through glycolysis down as far as pyruvate over the last couple of lectures, and now we're going to see what happens to pyruvate as it moves into the TCA cycle, the tricarboxylic acid cycle, or sometimes called the Krebs cycle, is discovered by Krebs many years ago. Not the first cycle that Krebs discovered, but maybe the most well-known one, also called the citric acid cycle. Before we start, I thought I'd start with a little outdoor biology. This is a picture taken by my neighbor. I have a second house out in Tucson, Arizona, and she got this picture of this guy at her water hole, which is about 5 feet off of her back porch. Okay, back to metabolism. And again, this is a diagram showing many of the reactions in metabolism. The dark vertical line in the middle representing glycolysis, and that feeds into this dark circle here, which is the TCA cycle, which is what we're going to talk about today. And you can see that not only does pyruvate feed into the TCA cycle, but many other compounds as well. Okay, so this is kind of the big picture here, showing two diagrams of the TCA cycle. At the top, focusing on the energy exchange, and the bottom actually focusing on the compounds that come in and go out of the TCA cycle. And so at the top, you see not only can we funnel carbohydrates through the TCA cycle, but fatty acids as well. And that's the main way to break down fatty acids. And also amino acids, some amino acids can feed into actually probably most, into the TCA cycle. The output then is energy in the form of ATP or GTP, and the total oxidation of carbon into carbon dioxide. So for each, as we'll see, 2-carbon molecule, acetate that goes into the TCA cycle, we get 2 molecules of carbon dioxide. So down at the lower diagram, we see some of the products or compounds that feed in or out of the TCA cycle. So the TCA cycle can, in addition to generating energy, it can also be used to synthesize certain precursors that can be used to build other compounds. The reverse of that can happen also. You can also increase the number of intermediates in the TCA cycle through the breakdown of some of these compounds. And these are shown here. You can see different amino acids from proteins feed in at different places. Aspartate on the left, some into pyruvate over here on the top right, some into glutamate at the bottom right. Again, fatty acids not only feed in through acetyl-CoA, but also through citrate. Okay, so very complex interactions going on between the TCA cycle intermediates and other compounds feeding in. But the main source of energy is all funneled through acetyl-CoA here, either from fatty acids or from pyruvate, which could be derived from sugars through glycolysis or from the breakdown of proteins and conversion of certain amino acids, alanine being the simplest example that's converted pretty directly into pyruvate. Okay, so we're going to center this whole thing around pyruvate as an initiator. And again, alanine can feed into pyruvate, lactate can feed into pyruvate. So if we have a situation where we've gone through glycolysis, generated pyruvate, and then converted that into lactate to generate more NAD. That lactate then, as we saw last time, can travel to other tissues, be converted back into pyruvate, where it can either go back and make glucose through gluconeogenesis, or it can send the pyruvate into the TCA cycle by converting it into acetyl-CoA. And so this is the major enzyme that converts pyruvate to acetyl-CoA, pyruvate dehydrogenase. So we're going to knock off one carbon molecule in the form of CO2, so it's completely oxidized. Also generate one molecule of NADH from NAD, and then that will funnel into the electron transport process to generate more ATPs. Okay, so, the other thing that can happen is we can convert pyruvate into one of the TCA intermediates, oxaloacetate, and this is a two-way reaction, I believe. So it can go back the other way. But the main pathway for generating energy is through pyruvate dehydrogenase, converting it to acetyl-CoA, and then acetyl-CoA combines with oxaloacetate to form citrate. And this is the structure of citrate. It's got 3 carboxyl groups, hence the TCA cycle or tricarboxylic acid cycle, and 1 hydroxyl group on this central carbon here. This is the structure of acetyl-CoA. Certainly don't want to memorize this, but just to give you an idea, here's the acetate over here that's going to get incorporated into citrate and then eventually be released as carbon dioxide. And this is the rest of the molecule that facilitates that reaction. And like a lot of other moieties that facilitate these reactions, they start with some kind of nucleotide here, in this case ADP. Now, I want to make the point here that the TCA cycle is located in the mitochondria as opposed to glycolysis, which is going on in the cytosol. So once pyruvate is formed, it has to be transported into mitochondria in order to proceed through the TCA cycle. Okay, this is the mechanism of pyruvate dehydrogenase, and again, let's not focus on the details here. Just want to show you that this is a very complex system that involves 3 different proteins, and that each of those proteins carries out a different function in converting pyruvate ultimately to acetyl-CoA and producing NADH as well. Again, that's whenever we see NADH, we think high energy because that can be used to generate multiple ATPs. So here's the entire cycle. And again, oxaloacetate combining with acetate, up here the red molecules, to make citrate. Okay, then first thing that needs to be done is to move the hydroxyl group on the citrate over 1 carbon. Okay, and that goes through this connotate intermediate, but it's really all one enzyme. So the next intermediate is isocitrate. At that point, then we can knock off the first carbon as carbon dioxide, and that then leaves alpha-ketoglutarate, which is a 5-carbon molecule with 2 um, carbo, uh, carboxylic acid groups, one on each end, and then as well as a carbonyl on the second carbon. So that's the reason for moving the hydroxyl group over one carbon. The next step converts alpha-ketoglutarate to succinyl-CoA, again losing the carbon, and now we've got two carboxylic acid groups in a 4-carbon chain, one on either end, one attached to a CoA, same CoA that was attached to acetyl-CoA at the beginning. Okay, and then the rest of this is just to get us back to oxaloacetate. So now we're down to 4 carbons at succinyl-CoA, and so we need to then go from actually we just need to oxidize this to put a carbonyl on the 2 position here, and so that's takes a number of steps in order to do that. And those steps, as well as oxidizing the succinate to oxaloacetate, since that's an oxidation, it's also going to generate energy in the form of GTP in the conversion to succinate, and then FAD, which can also function in the electron transport to generate ATP, but not as many ATPs. And then finally an NAD, and then the final state from malate to oxaloacetate. Okay, so again, this shows the cycle. We've got NAD formed at 3 different steps here, plus an FAD, loss of, I shouldn't say loss, but generation of 2 carbon dioxide molecules. And somewhere in this mess, there's an ATP formed and a GTP. Okay, just look at a couple of the steps in a little more detail briefly. Again, we're not going to spend our time memorizing all these intermediates in the TCA cycle. If you ever want to know what they are, you can look them up within a few seconds on the internet. For those of you that have a strong background in chemistry, this is probably going to make sense to you. You know, my background is really in chemistry. I can pretty much piece together the TCA cycle, even if I haven't seen it in 10 or 15 years. But I don't know that that's necessary here. Okay, so again, once citrate is formed, the next step is to move that hydroxyl group on the third carbon to the number 2 carbon to make isocitrate. Now there's a pretty potent inhibitor of this, and the compound that is generally added is this fluoroacetate. So it's replaced one of the hydrogens with a fluorine on the acetate molecule, and that gets incorporated into acetyl-CoA as fluoroacetyl-CoA. So acetyl-CoA synthetase recognizes it as acetic acid and adds a CoA to it, and that becomes a very powerful inhibitor of aconitase, a first step in converting citrate to isocitrate. Okay, this box again tells you a little more information about that. I'll let you read that on your own. The interesting concept here is this idea of a suicide inhibitor, and that's something that's not toxic by itself, but when it's ingested, it gets converted by the organism into a toxic compound. Okay, another reaction I just want to talk about briefly is conversion of alpha-ketoglutarate to succinyl-CoA by alpha-ketoglutarate dehydrogenase. Interesting thing here is it's very similar to the enzyme that converts pyruvate to acetyl-CoA. And so when you look at the structure of these two proteins, you find there's very similar homology at the active site of these enzymes, where the the part of the protein that recognizes the substrate will be different because that one's going to recognize alpha ketoglutarate and one's going to recognize pyruvate, and that's a common concept that we see very often, that these enzymes that carry out the same reaction but only on different substrates um, have active sites that are very similar to each other and probably derive from the same source. You may talk about that a little more when we talk about gene structure. Okay, so the next step is to convert succinyl-CoA into succinate, so just take off the CoA. And in doing that, we generate or convert an ADP to a GDP into a GTP, so we generate some energy there. And this is sometimes called substrate level phosphorylation because this reaction can actually generate triphosphate nucleotide directly instead of going through like the electron transport or some other process. The other thing to remember is once we've generated GTP, that can be readily converted into ATP because these nucleotides can swap phosphates readily with no change in energy. Okay, so back to the original diagrams here. Again, when we look at the energy diagram at the top, what we see again, we're feeding acetyl-CoA into the TCA cycle, and that can come from carbohydrates, fatty acids, or amino acids. And once we go around the cycle, we generate 3 molecules of NADH, a molecule of FAD. These both feed into the electron transport system to generate ATP. And we also generate 1 GTP, and the result is the total oxidation of 2 carbons into carbon dioxide. So you can see that, when we talk about glycolysis, not generating a whole lot of energy there, but it generates it very quickly. And so if we need quick energy, that's the way to go. But the TCA cycle, much more efficient at generating energy, or generates a whole lot more energy per carbon than the breakdown of sugar into pyruvate. Now, there are many organisms, particularly bacteria, that don't generally use the TCA cycle. If they're in a situation where there's plenty of substrate, then you might call them lazy. They just will break down sugar into pyruvate and toss out the remaining molecule as lactate. And many of the bacteria that have been sequenced now, and their genomes sequenced, and there are thousands now, are missing or seem to be missing enzymes in the TCA cycle. But most of them have some of those enzymes, and it's not clear why they would have some and not others. They don't appear to be able to carry out a fully functioning TCA cycle run. But they may be used to generate other intermediates. They may also take up intermediates from their surroundings and feed them into the reactions that they do have enzymes for. So even though they may not complete the whole cycle, they clearly can do part of it. Okay, again at the bottom, focusing on the different molecules coming in and out of the TCA cycle, and um, many molecules, as we talked about, can feed into the TCA cycle. Uh, and those TCA precursors can be used to make a lot of different molecules. So most of these are two-way reactions. The one that's particularly important here, that's a one-way reaction, is the conversion of pyruvate into acetyl-CoA. Okay, so you can't go back directly backwards here. So what this means is that we cannot feed fatty acids into acetyl-CoA and then convert it to sugar. So you can't convert fats into sugar. Convert sugar into fat through acetyl-CoA, but you can't go the other way around. So if we take in an excess of fat in our diet, we have no way of converting that to sugar. Okay, here's then the full reaction. Again, acetyl-CoA coming in, generating 3 NADHs, 1 FADH. Those feed into electron transport or oxidative phosphorylation to produce ATP. And then we've got 1 ADP converted to ATP. And the output then is 2 carbon dioxide molecules. And that's the overall reaction shown at the bottom here. Okay, here's the spreadsheet, I guess, the tally for energy generation. And this is starting not just the TCA cycle, but starting with glucose. And again, as you recall, to get things rolling, we have to expend 2 ATP molecules to phosphorylate the glucose, and then fructose at both ends of the molecule. After that, we generate 2 NADs, one from each of the 3 carbon molecules. Now, if we're going to convert pyruvate into lactate, then those get converted back into NAD. But if the pyruvate's going to go into the TCA cycle, then those NADs can go into the oxidative phosphorylation and generate ATPs. And there's a little variation in how these things are shuttled in and out of the mitochondria, and that can affect how many ATPs are formed. Okay, then as we proceed into the TCA cycle, we generate a number of ATP and NADHs. So the final tally here is the generation of 30 to 32 ATPs for every glucose molecule. Now remember, we're starting with glucose with a molecule that's pretty far oxidized. You know, there's an oxygen on every carbon. So, you know, if we start with fats, which are totally saturated, we can generate a lot more energy per carbon than we would with glucose or other sugars. Okay, so this is a simple diagram then showing all the intermediates in the TCA cycle. And this also shows places where the process is regulated. As you might expect, it's regulated at the front end on the conversion of pyruvate to acetyl-CoA, and then also regulated in the conversion of isocitrate to alpha-ketoglutarate, and the next reaction to succinyl-CoA. And as you can imagine, if there's a large amount of ATP in the cell, you don't need any more energy, then that's going to slow down this process. Same with excess of NAD. So for the most part, regulation is pretty much what you'd expect. There's also allosteric control. In a number of different places, by a number of different compounds, and this may vary from tissue to tissue, but you can see here insulin is stimulating the process, as well as calcium, probably two big regulators of carbohydrate metabolism, and there are others as well. Okay, this is the last slide. Is some significant concepts about the TCA cycle. It results in the complete oxidation of acetyl-CoA to carbon dioxide, with the generation of several ATP molecules after the NADH that's generated goes through the oxidative phosphorylation pathway. It's considered the final common oxidative pathway because all of these different molecules, carbohydrates, fats, proteins feed into it, and the endpoint is a totally oxidated carbon dioxide. It also serves as an integration of major metabolic pathways, and we saw that. We've got all these molecules coming in through the cycle. We can also use those intermediates to generate different amino acids as well as other compounds, and those often are, or more often than not, are two-way reactions, so we can generate new intermediates in the TCA cycle if we're running low on them by feeding in those molecules at different points along the cycle. Again, fat can be digested or metabolized into carbon dioxide, but it can't go back and make carbohydrates. However, an excess of carbohydrates can be converted into fat, and we'll be talking about that later on. And no net synthesis from carbohydrates from fat. And one last thing, the carbon skeleton of amino acids can enter the TCA cycle. Okay, that's all I'm going to say about the TCA cycle. It's pretty straightforward. We don't need to belabor it. This last slide pretty well sums it up, although there are probably a few other key points that we need to remember. We're going to move on then to the next topic, which I think will be the metabolism of amino acids, both the breakdown of amino acids and generation, synthesis of amino acids. So see you next time.